Okay, Assalamualaikum, selamat petang. Selamat datang semua. Kita ada dah 30 lebih orang, so Alhamdulillah we can start now. Saya pertama kali nak ucapkan terima kasih kepada semua yang hadir, tahniah beserta amal yang menerima cabaran, dan ini kali kedua insyaAllah kita cuba adakan tiap-tiap bulan untuk kita bincangkan isu-isu berkaitan dengan biodiversity. So I understand there Sule from Nigeria also here. Welcome, thank you very much. Expected from Qatar, Nigeria, and Maldives to join us. So for the universities, for the lecturers in biology department, maybe you can ask your students from overseas also to join our meeting, monthly meeting, to discuss about the issue of biodiversity. Just a mukaddimah, biodiversity is very important. I just have a meeting today in the morning, start from 8:30 to 10:30. We discuss about the solid waste management, how it is affect the biodiversity, especially on the aquatic environment. And it's a lot of discussion, and then they reverse back to the issues of the management and administration. Then after that, I was very lucky, was invited to meet this staff from French embassy. And another thing, they are talking about the biodiversity. We have been doing or collaborating together with French scientists for many years, but not well highlighted. So I took the opportunity this morning to highlight all the activities and then our collaboration with them, and they offer a lot of things to work together on the biodiversity. And then Malaysia as top biodiversity in the world, top 12 biodiversity in the world. Then they look at our priority in the government and politically and economically, accessibility and so on. They are so interested in collaborating with Malaysia. So I hope we can find out how to work. I will propose to them, I already proposed to them also, so that we need a central collaboration. One reason why we fail in working on biodiversity is our networking and centralization or coordination within the researchers on the issue. So I try to work on this networking, so that people can be joined together. Imagine if Sule in Nigeria, I can link Sule either with French scientists in Nigeria, in France, and British scientists in United Kingdom to work together with Sule in Nigeria and collaborating with scientists in Malaysia. So this is all about. With a new networking, is everything is possible. So I hope this seminar meeting will promote this idea of biodiversity and conservation, which is we are talking very, I cannot say aggressive yet, but we show some interest, what many my counterpart in the world are talking. And okay, I will elaborate it more, but today I'll see how. Today we have two speakers. Number one, Dr. Izzati from UPM, from Biology Department, and then Professor Rusea Go also from Biology Department. Our focus today is on last month we talked about fish. Now we move to another topics, fungi and orchids. We will find many experts. There are lists of experts approach me to contribute to this short discussion, because I said this is a santai, this relaxed, no pressure. You don't have to present slide if you don't have time. What I need is your experience and experience and knowledge to give you ideas and input, so that we can promote more young scientists to be to develop their interest in biodiversity study. All right, I don't want to to start talking because this morning I talked a lot already, and then I occupied most of the discussion as usual. Why? Because we want to make it happen. We don't want just a theoretical and a lot of things, conflict between the policy makers and then the education people, including the academician. So we don't go beyond so that we want to see things happening. But, biodiversity is not many people. So if you talk about fungi, maybe how many? Dr. Ain akan menceritakanlah sekejap lagi. Alright. So now who going to start first? Dr. Ain or Dr. Iza? Dr. Rusea? Prof. Rusea? Right, I didn't see Prof. Rusea here, so I think Dr. Ain will start first. I give you the 20 minutes to speak, and later maybe we can have question and answer. Dr. Amal, where are you? I didn't see you.
I'm here.
Yeah, okay. Dr. Amal will look on the chat box, so in case whatever you want to do with chat box, sometime you want to register your name, you want to ask a question, and the Tama will be assistant to me. And maybe next time in the poster, put a picture of the Tama as a co-moderator. Sule smiling. All right. Okay, Dr. Ain, you want to start first now for 20 minutes?
Okay, thank you, Prof. Ahmad. So, okay, I will share my slide presentations. Okay, can you see my slide?
Yes.
Fusarium,
Yeah, we have here together with me, if I'm not mistaken, Dr. Nick from USM. Yeah, he very expert in Fusarium, especially Proliferatum and Verticilloides. So we have also my former FYP students, graduated 2010, Dr. Razik. Yeah, now Dr. Razik in UIA, lecturer in UIA. So thank you for joining us. Okay, so thank you, Prof. Ahmad. So today I would like to share a bit on Fusarium species, about the Fusarium species. When we talk about the diversity of fungi, so we have a macrofungi, we have a microfungi. So even if you give me one day, so we cannot finish to share all about the fungi world. So for this afternoon, I will share about the diversity of the Fusariums, ecological and their industrial importance, because usually when we talk about the Fusarium, then we always say that the Fusarium is a common plant pathogens. Yeah, as a living organism, microorganism, they have a bad and good effect of the fungal, and also even other organism also, they have a good and bad roles in our nature and also in the community. So this is my overview of my sharing this afternoon, about their classifications. So at least you know a bit what is the Fusarium and how it looks like, and then the diversity and the distributions, the ecological and importance, and also their importance in our industry. So because I believe that most of you that's working with fungi, there's no this Fusarium that's comprise a diverse of the species that can cause a disease, and also they are very good as a saprophytes. Okay, so for the classifications, the Fusarium under Ascomycota. When we talk the Ascomycota, a bit technical. So when we talk about the Ascomycota, so most of the members under Ascomycota, they produce the perfect stage. So the name of the Ascomycota, because they produce this ascocarp, that's a fruiting bodies. So in the Ascoc card, they have a ascospore. So Fusarium together with Coletorchicum, the glamour genus, the genera, and also the Trichoderma under Sodariomycetes, yeah, and then the class of the Sodariomycetes. So this Fusarium very large genus, filamentous fungi. So filament means that they have the hyphae, yeah, that's have the septate as well. So most of the species that harmless saprophytes and abundant members in soil microbial communities, and usually you can find them in a soil, yeah, but they also very good saprophytes and associate with disease plants. Even they are non-pathogenic, but they associate as saprops. Okay, so based on their diversity, yeah, years by years, actually the numbers of the species under Fusarium that increasing, and recently more than 200 species. So this because of several species that belongs to this genus, they characterize based on the morphological, physiological, and ecological characteristic. Even we have the phylogeny species, yeah. So they use the sequence, genetic sequence to group them into new species. So and then also we have also the biological species, yeah. So that's the reason they are very diverse. And in terms of the habitat, yeah, the Fusarium, you can actually isolate them from marine, freshwater, or soil. And even if you have the agar at your home, yeah, you just open for 10 minutes and then you close them and then you incubate for 24 hours or maybe 72 hours, then you can find other than aspergillus, you can also have the culture of Fusarium, because they have everywhere. Yeah, this Fusarium, aspergillus, penicillin, everywhere. We can actually isolate them. So in terms of the morphological characteristics also, they are very diverse, yeah, based on the size, the shape, yeah. So even the shape. So for example, they have a microconidia. Later after this, I will show you how it looks like, the microconidia and also the macroconidia. And the shape also, they are very diverse. They have the globus shape, and then they have a truncate, ovoid with truncate shape, yeah. So, and then also nowadays, because of their morphological characteristic very diverse, yeah. So various in terms of size and shape. So they use also the metabolites profiling, yeah, and also the molecular techniques to identify the species. In our lab, mycology lab in Department of Biology, we identify Fusarium species usually based on the morphological characteristic and also the gene sequencing, and using some of the specific primers. And also we, if let's say that the culture that we targeted is under Fusarium fujicor section Laciola, so we will proceed for mating population as well. Yeah, because of this variability in the species, so that's the actually they relate with the ecological conditions. Yeah, so okay, like I said just now, it's very hard to identify the Fusariums. Yeah, you just imagine we have more than 200. Nowadays we have more than 200 species. Yeah, even I just give this example. Yeah, 2 species. Yeah, that's the reason when someone that's bring the plates of the fungal isolate, yeah, we cannot actually identify based on our naked eyes, based on the colony features, based on the morphological characteristic itself. We need to identify them, use the alternative technique. But morphology very important. First, we need to observe all the morphological characteristic, and we undergo the molecular markers, because you can see here based on the plates, yeah, they have a similar characters. For example, Fusarium proliferatum and verticilloides. So the only The only characteristic to differentiate them is actually the phyllite that produce the phyllite. So Polyphyratum that produce polyphyllite, for example here, I believe that some of you this is first time that see the microscopic characteristic of fungi. So this is the microscopic characteristic of Fusarium species. They produce microconidia. So the microconidia have a various shape. So for example, they have the oval shape, and then oval shape with truncate base. Because of this, for example, here is microconidia in chain form, like this chain form. So that's the reason they have a truncate base. And then we have also that's a microconidia have a piriform shape, and also the void and global shape. So you need to observe this, and to having a very good and standard size, standard shape of the conidia, the microconidia, macroconidia, you need to culture on the specialized medium lah. For example, that's carnation leaf agar. You cannot actually observe the microscopic characteristic on potato dextrose agar. So we have actually special medium that actually they will produce form a very uniform and standard size shape of the conidia. This example of macroconidia, in terms of the size also, they have a various size of the macroconidia from 1 septate to more than 4 septate, which is contain 5 cell for one macroconidia. So some of the species produce microconidia in chain. For example, here this is the chain of the microconidia that produced by Fusarium proliferatum, Fusarium verticilloides, and then this example the false head. For example, the Fusarium subglutinus, they produce the microconidia without chain, so they produce in false head. But those species that produce microconidia in chain also produce the false head. Okay, and then this is the famous species, Fusarium oxysporum. When we talk about the Fusarium oxysporum, then people cross in their mind. Usually that's Fusarium wilt disease. That's caused banana Fusarium wilt disease in banana, also tomato. Is common pathogen for our plants, crops. So, and special about the Fusarium oxysporum, they have the forma specialis, which is the host specificity. For example, that when they infect the banana, we call as Fusarium oxysporum forma specialis cubense. When they infect the melon, so we call as a Fusarium oxysporum forma specialis melonis. So, and then this example of the chlamydospore is not all the Fusarium produce chlamydospore. So that's the reason we usually when the crops infected with Fusarium wilt disease, so it's hard to cure them because of this chlamydospore. Can actually survive in our soil for 30 years old, 20 years old. So that's the reason you have to rotate your crop. When your crop having a problem with Fusarium wilt, then you have to rotate with other crop that's resistant to the disease or is not suitable to the disease. So, okay, other than that, based on the biodiversity, they also, this Fusarium also have the perfect stage. So, for example, here is the perfect stage of the Fusarium verticilloides. The perfect stage of Fusarium verticilloides, they identify and classify as a Gibberella monoliformis. So that's the reason usually in Ascomycota, that's very special classification. In Ascomycota, they have the perfect stage. So, and usually the ascospore, the ascospore of the perfect stage of the Fusarium actually bigger than conidia for the anamorph stage. Okay, so about the distributions, yeah, because when you Google Fusarium, you will get extract abundance of article about a Fusarium species. So this example of the Fusarium graminearum. Fusarium graminearum is actually a very famous temperate species that can cause, for example, head blight of the maize, wheat, and also barley. But now it's scattered all over the world, because of the climate change, because of the adaptations, survive, so they actually can survive. In Malaysia, so actually my group, during Dr. Razik, Dr. Razik did his FYP, some of his senior that's isolated Fusarium from maize. We successfully isolated Fusarium graminearum from maize sample from Cameron Highland. But because of this Fusarium graminearum actually low temperature isolate the species, so that's the reason actually the isolate cannot survive before we keep and store them. So we just realized that we have a graminearum, when we do the slide culture, and then later they die, so because of the temperature. So but we have the graminearum, we isolated from Cameron Highlands. So and then this actually the effect of the climate change as well, the temperature that's increasing. So about the Oxysporum, you can see here. So the Oxysporum that's like I said just now, is very famous. They scatter all over the world, even in temperate area also. You can find actually the Fusarium Oxysporum. So so this same as the Fusarium solani. Yes, Fusarium solani, that's famous for the soil-borne. Same with Fusarium Oxysporum as well. So you can see that they all over the world. We can find the Fusarium solani, Oxysporum, because they are susceptible. And even they actually they are soil-borne, so that's very easy for them to survive. Okay, so based on the ecological significance, like I said just now, I share much about Fusarium as a plant pathogen, and then they are the most species that usually as a seed-borne, they are very typical genus and well known as a soil-borne genus that usually I share just now about Fusarium Oxysporum and also Fusarium solani. Actually, there is many species that actually belongs to this soil-borne pathogen. So and then but in good side of the Fusarium, like others organism, it's not all isolate under the same species as a pathogenic. For example, just now I share about Fusarium Oxysporum, okay, plant pathogen, but only 60 to 80% that's when we test it, usually they can cause a disease. Can cause a disease infected the crops. But the others, their roles usually as a saprophytes. So they are non-pathogenic. So this is the non-pathogenic, you can use as a biocontrol agent. But of course, because in disease pathogenesis, that disease affected the triangle, the three factors about the pathogen, the host, and environments. So all these three factors actually affect the pathogenesis. So if let's say that the pathogens is a pathogenic but the host is resistant, so they will not cause a disease. But in Fusarium oxysporum, even you have, for example, you have 100 isolate, so it doesn't mean that you identify them into the Fusarium oxysporum, they confirm as a pathogenic. No, you need to test it them, and usually based on our findings, yeah, if let's say that we aim for Fusarium oxysporum cubense, yeah, only 40% that's cause a disease and pathogenic. The others, they are non-virulent. So this usually this non-virulence that we can use as a biocontrol, we can use as a candidate for bioremediations. Yeah. So out of 200, more than 200 species, yeah, for example, Fusarium semitectum, yeah, usually they not non pathogenic species. So they give a good effect as a saprophytes, yeah, they decompose, yeah. So and then for example, Fusarium compactum, Fusarium chlamydosporum, yeah, Fusarium chlamydosporum actually in 2008, if I'm not mistaken, that's we isolate Fusarium chlamydosporum from orchids, yeah, as a endophytes. So they are not pathogenic. So and then we also have the Fusarium equistite. Yeah, so all these species actually is not pathogenic. So many other species that actually is not pathogenic. So yeah, that's if let's say that after this, yeah, someone that's the young lecturers, young researchers that's interested to working with Fusarium. If let's say that your specialty is on the bioremediations, yeah, biocontrol agents, so you can actually use as one of the fungal, yeah. And but of course you need to test to do the pathogenicity test before you claim that the isolate is non-pathogenic. Okay, so I share you that's what we have done, yeah, because I'm start working with Fusarium since 2000, yeah, dengan Prof Baharudin Saleh and also late of Dr. Farida, so Prof Farida. So this very famous, I believe that some of you did working on this disease, yeah, Fusarium wilt disease, of course. Very serious disease in Malaysia as well and worldwide. So caused by Fusarium oxysporum, Forma speciale cubense. Fusarium will attack the vascular system. So that's the reason when they colonize this phloem and xylem, so the photosynthesis cannot occur and no food. So that's the reason the leaf turn to yellow, yeah, and wiltings, and they will collapse later. So and then this is common, yeah, the symptom that I believe that most of you maybe this afternoon got the goreng pisang nanti, yeah. So check your banana, yeah. So is there any symptom of this? So actually this fruit rot of banana, the pathogen of the fruit rot is have several species, yeah. It's not only Fusarium, other filamentous fungi also, the Ascomycetes fungi also can cause a fruit rot banana. My student, Baiti, yeah, she working with fruit rot during her master's study. Yeah. So actually, what you can do is actually if let's say that you brought fruits or vegetable, so please make sure that your fruit you keep store well your fruits or you consume directly. Yeah, don't keep and store for longer because of it will give chance to this Fusarium accumulate, yeah, and colonize the fruits. So because the bad things is when they produce the spore, then they will produce the toxin. So before they colonize, so please make sure that you eat that. Okay, so and then same also this Fusarium will on cucurbits. We did on the watermelon, yeah, cucumber, melon, rock melon as well. And then this fruit rot or Fusarium wilt caused also by the Fusarium oxysporum, but Forma specialis lycophysi. So like I said just now, Fusarium oxysporum have a Forma specialis. Just now Forma specialis cubensis. When we test it on tomato, so they will not cause a wilt disease. So this Fusarium is host specificity. So for example here, for Fusarium oxysporum Forma specialis lycophysi, when we test on the banana, they are non-pathogenic to banana. So and then this one Fusarium earroot, so caused by Fusarium proliferatum and verticilloides. We also, this is based on our study, but actually many species of the Fusarium can cause a Fusarium earroot. Even Graminearum also can cause a Fusarium earroot. And based on our study, in 2010, when we do the samplings, corn meal sample, and most of the sample is contaminated with Fusarium proliferatum and verticilloides. Other than Fusarium, Aspergillus and Penicillium also contaminated. So, and usually it's not difficult to get them. So you just imagine if our animal feed stuff that contaminated with this microorganism, Fusarium, Aspergillus, Penicillium, so they will colonize and produce the toxins, and we feed our chicken and everything, so they will bioaccumulate if we
Ain banyak lagi kan?
Oh, tinggal berapa minit?
Habis lah.
Nak habis dah? Okay, saya jump to, okay, I jump to the good effects of the Fusarium. Okay, so like other microorganisms, they have secondary metabolites. So I talked many about the mycotoxins, about the aflatoxins, fusaric acid, all those things. But I'm interested to share, this is actually the effects of the toxin. So I just want to advise, remind me as well. So we need to avoid to eat the contaminated foods. If we see there's a have lesions and the fungal, so we need to avoid to eat them, because this toxin is bioaccumulate. They will remain in our body. They bioaccumulate in our liver, our kidney. So for 10 years, 20 years, so who knows that? Because it can cause a liver and kidney cancer as well as others health disease, health problems. So the other good thing about the Fusarium, so because we know that they have they produce the sporulation, they produce the secondary metabolite during the sporulation stage. For example, here they have they be able to produce the enzyme. I just list some of the enzyme that produced by Fusariums. So there is many other enzymes that are produced by the Fusarium, and actually it's good for the industrials. And then another is about the bioaroma. That's a secondary metabolite that produced by this Fusarium, and also the pigments. Usually these pigments, they use for dye industries. If you want to use for the foods and also for the medical industry, so you need to test many things, because of some of the pigments. For example, that Aurofusari, they are toxins, so you need to test it. If let's say that you are going to extract the pigments and use for food and medical, but usually nowadays they use for dye industry, for the dyeing process, dye process. Okay, I think you can see here, there are diverse. You can actually extract the pigments. But be careful when you, even it's not only in Fusarium, so in other organisms as well. When we want to purify the metabolites, then we need to test many things. Okay, that's all. Thank you, Prof.
Thank you, Tuan. Sorry, we planned for 1 hour relax and sembang-sembang. So I'll arrange another time that Amal will arrange, because we have a series. We have monthly talk to promote the biodiversity. I can see many people asking when. Still now they didn't see the date. Maybe we need to put the date bigger, Amal. uh, so I think we proceed with the next one. Prof. Rusea is here already. Prof. Rusea, so you can start now. Try to get it in 20 minutes, so we can fulfill our promise. Okay, Prof. Rusea?
Okay. Okay, can you see my presentation?
Can, Prof.
All right.
Mute my, okay, I'm talking
Okay.
alone. All right, thank you. Continue.
Okay, let me start with the, I was given this topic of biodiversity and conservation in times of pandemic, and for my group of team, we actually studied on the orchids and also on medicinal plants. Before that, I would like to welcome all that is online today. And I know that this presentation is not something that is a concise kind of presentation, and to share of what are the things that have been that I have been doing or that is been done during this pandemic. So I'm sharing with you how we do biodiversity conservation, and during this pandemic, you all know very well that we can't go out to the jungle where that is the place where we are is our major playground in the forest. So during this pandemic, we are tight, and we're in home arrest that whereby we cannot go out most of the time, and Selangor being the red zone for during the whole duration of pandemic. So knowing that so well, we have to opt to something that will make our existence as researchers or what you call it worthwhile as well as productive. So as we all know that the COVID-19 pandemic, so how it comes about and what is the cause and so on and so on. We know all about this. We are aware of what the SOP that we are supposed to follow, and as well as what are the things that the government has been proposed for us to do. But at the end, the figures still rising. So the highest that so far that we have seen is the 22,948 cases per day, as compared to our number of population that we have in the country. This is very, very high, probably the highest in the world, because this kind of infection is does not occur in the country where population is very high. So let's say for India, now we are so far higher than India in terms of what you call it, infection. But no matter what, research got to continue. This kind of thing, we cannot take this as an hindrance to our work, to our research, and to how we can share what our research findings are all about. Okay, so this is just roughly that to share with you what is pandemic all about, what we have done, what are the things that we know. And definitely, I'm sure all of you have your second dosage by now. And for myself, I have a second dosage about a month ago, and I hope that the immune system in me has been developed, as well as all in you all, and I hope that all of us will continue to protect ourselves from this virus. As we know now, there's a new Delta Delta variant, which is so much what you call it, causing too much death and rapid kind of infection. So please take care. And before I continue, that I would like to just pray for all of you to be in safe hands and be protected all the time. As for what I did, okay, there are three main things that I do during this during this pandemic. I also was given my last sabbatical for my entire employment in UPM. So that was my 9-month sabbatical from October until 30th June 2021, October 2020 until the 30th of June this year. So all I do is I was given a fellowship to go to Leiden, but I cannot do that as well. I was supposed to go to Kew and doing all those research that I'm supposed to do, but all those are cancelled due to the pandemic and the restriction of flying and so on and so on. And then then when you are confined to your house and not even allowed to go out to the jungle nearby your house, so what you need to do. So this is what I plan and I have been doing in terms of orchid research, in terms of my CRIP project, the Korea Research Institute Bioscience and Biodiversity collaboration project on medicinal plants, as well as what I've been doing in terms of book writing and also journal papers. Okay, so in terms of orchid research, I have this year alone, I have finally registered 4 of my new hybrids that I did together with the nurseries. This is an industry kind of collaboration work. So this is not done by yesterday or 2 years ago. This project started in the year 2000, as early as 1999 actually, and finally the hybrids comes with into stable clone, and we can now register some of the hybrids. So these are the 4 new hybrids that been registered early this year. I have one named after our Sultan of Selangor, which is named Pavillonanda Sultan Saifuddin Idris Shah. That was already been registered as well. And then I have another one that is named after the Permaisuri of Selangor as well. This is known as Pavillonanda Nanda Tengku Nur Ashikin. And another one is named after the former mayor of Shah Alam city, which is known as the Bangkuante Haris Kasim. And finally, I give the honor of one new hybrid in conjunction with UPM 50th anniversary celebration. I named this hybrid as Aranda Uniputra Gold. So this kind of work is not an overnight kind of a success. It has to be something that you work closely together with people who actually are able to take care of all the clones and all the hybrids that you together created. So as for UPM, we don't have the facility to have this luxury kind of thing to have and to keep and also to create this kind of environment where we can create hybrids like this. Because we don't have the passion, we don't have the manpower to take care of all the clones, the new hybrids that we create. And then in terms of apart from that, since we are very easy to join any conference, international conference. For that particular reason, I presented in orchid conservation in the World Orchid WOC World Orchid Conference that was supposed to be held in 2020, but because of the pandemic that broke before, just weeks before the conference. The conference was then changed into a virtual conference in 2021. So the World Orchid Conference was actually virtual conference, and basically being being hosted by Taiwan. And then I was called as a keynote speaker for the first international conference on orchid in Iran. So that was done in June this year. So what I shared with them during this conference is all the things that mostly all the what you call it, the achievement that we've been throughout the orchid research in what I've done with my team so far. Okay, so during that orchid what do you call it, the World Orchid Conference, we present the paper, and as well as in the first International Iran Orchid Conservation Conference, I basically present papers that are being co-wrote with my former students and current students. So what I shared with them is about what we do, what are the orchid diversity, the conservation strategy, and conservation initiatives. So most of the things that I shared with them is something that what is the current update in orchid study and orchid research, as well as orchid conservation initiatives. So this I shared with them how we go to the field, and with a luxury of sometimes with helicopters, and not to mention the dangers of going through rivers that are the roads that is actually across the rivers with literally with water. And of course, this is one of the trip to what you call it, Taman Negara, whereby you have to cross 13 rivers like this in order to reach the peak. And finally, we reached the peak. So this is one of the memorable trip that I made, whereby I almost lost my life during the time that we are going back from the trip, because the river was actually a swelling river, and it almost swept me off to death. Okay. And of course, we do the orchid conservation in many parts of the country, and with a collaboration with the JPSM, Jabatan Perhutanan Negeri Semenanjung Malaysia, as well as the state forest departments, we managed to carry out some study and some collection and some records for in advancement of orchid studies in Peninsular Malaysia, and as well as in Sabah and Sarawak. Okay, some of the collecting tips are how we do our collecting, and how those are the people that's around me that we helping me to collect all those samples, and as well as identify new locations for study. Okay, so these are the data collection and how we do the data collection and how we do the sample processing. And this is basically what must be done when you do on biodiversity study. This is only on plants, but on other organisms also we have to do the same thing. Okay, for orchid diversity, we wonder how many species. We will never know how many species we have until we study the whole area of Malaysia. So as far as it's concerned now, we are roughly about 4,000 species, and we have only a land mass of 330,000 kilometers square. So the number of species that's found within this area, we are considered as one of the biodiversity hotspots for orchids. Since as far as 2020 is concerned, until 2020, there are 202 species are identified new to Peninsular Malaysia. New to Peninsular Malaysia doesn't mean there are new species yet. They are not new species, but they are new records to Peninsular Malaysia. And we have 228 species which are endemic to Peninsular Malaysia. And orchid highest diversity, we identify the area in the mountain forest. And Mount Kinabalu itself, okay, the record and the diversity that was reported by Wood in 1993 is already up to 711 species in one mountain alone. So this number definitely is not conclusive. I mean, not the actual number of species that is found there. This is just an estimate and what has been seen so far. And I'm hoping that in a few years to come, that's new. I mean, a few in a short moment, a while, in a few years to come, we will update the number of species found in Kinabalu. I'm sure of that. Okay. The Malaysian orchid species with the number increased close to 4,000, accounted to 16.7% of world's orchid species. So that is not little. So here are some of the tiny, elusive kind of orchids that we found, and they are very beautiful orchids we found in Malaysia. These are all endemic species. We know them as corybas. They are as big as, the biggest would be about a size of a 20 cents. Malaysian 20 cents, and these are the three endemic species that are actually found in Peninsular Malaysia. We have Corybas puati, which is actually a new species, Corybas langorensis, endemic to Selangor, as well as Corybas villosus, endemic to Peninsular Malaysia, in three different most of it found in the main range of the Peninsular Malaysia. So Orchid Conservation Initiative in Malaysia, this is the presentation that I did during the World Orchid Conference. So most of the things that I discuss here is what we have been doing, or what has been done to alleviate the studies of conservation of orchids, and also how what has been done so far. Okay, so of all, we know that the major threat to orchids and other biodiversity is deforestation. So this is something that is very alarming and very disturbing. As for Malaysia, is the highest in terms of deforestation among the many biodiversity hotspots. Okay, as far as since 2000 to 2012, okay, there are about 14% of our forests been cut. So this is something not really encouraging for biodiversity, and also for all those conservation, biodiversity conservation researchers, and also NGOs. And what we do is in order to what you call it, to have a better understanding of the orchid diversity, we found out that by going into the active logging concessionaries will help to find new findings, something that we never discovered before. So this initiative, I started with the Forest Department of Terengganu, whereby this is the things that we went through, the newly or the just cut logging blocks. So we found out that new discovery through this particular work that we did through rescuing the orchids from the logging concessions. We found out that there are 53 additional new records for Peninsular Malaysia. We have 17 new records for Borneo, and 7 new species were discovered, and endemic to Peninsular Malaysia were now increased to 215 species. Species new record to Peninsular Malaysia, these are found during my study in Perlis. So many of the species found as new records in Perlis are the elements from the southern Thailand. So here you can see that Gastroculus hainanensis basically first found in the Hainan Island that is just south of China. And we have a Gastroculus obligus and Phalaenopsis amboinensis was previously thought to be only endemic to Ambon, but found in somewhere in Pahang. And we have Hymenorchis javanica was as well as known as endemic only to Java, only in one mountain in Java, but we found this in our in Pahang as well. And of course, the new records to Peninsular Malaysia like Didymiplex Siopsis of Kironensis, this particular species was found in Hainan Islands, but also found in Langkawi. And Dendrobium sianal, also a new record found in Langkawi. And Liparis geophila found in Gunung Jerai. Bulbophyllum sigaldiae also found in Gunung Jerai. All these are the element from the north and also from Sumatra. Okay, new species recorded to Peninsular Malaysia continues with Area Ocracea, and we have Trichotosia densiphylla and Trias intermedia, as well as Trichospermum pensile. Area and Trichospermum pensile are more discovered as new records to Perlis. Trichotosia and densiphylla and also Trias were new records discovered in Pulau Langkawi. And many of these, like Crepidium, Aculatum, okay, and also the Dendrobium agamanse, Liparis vestita, most of these are new records that was either from Sumatra or the northern or Thailand.
Banyak lagi ke, Prof. Rusea?
Banyak lagi, sekejap je lagi. Ni baru 5 minit, Pak Pemat.
You tak
Discovered
tengok
after
jam.
15 years.
Jam,
Okay.
jam you mati kot.
Memang. Okay, Macropodontus, this is discovered after 115 years. Okay, so these are the, okay, tak lama lagi, tak banyak lagi.
Okay, dah, go.
The new species or to science, these are among the new species to science. This was named after me, Dendrobium Rusea. And this is named after Datin Paduka Dato Aini, Dendrobium Ainie. And this is named after the Sultan of Terengganu, Dendrobium misaniae. And we found new Bomhidia found in Petuang in Terengganu. It's named after the place. We call it Bomhidia petuangensis. And of course, vanilla. This is something that been talked about. One is Vanilla nur ashikiniana, endemic to Terengganu as well as Selangor only. So far, we only found it there. And Vanilla sanguineo venosa is also found only in Perak and Pahang. And of course, the new species, Corybas poati, found in Kedah. And the splendors of Malaysian orchids, these are some of the pictures of plants, some of the posters that have been created to highlight what are the species that have been found in Peninsular Malaysia, as well as what are they all about. And they are most of them, although they are small flowered, but they are beautiful and with scent. And some of them, just show you some of the pictures. These pictures are actually from, these posters was actually created by Dom Nikong from Terengganu. He's an avid conservationist and also explorer, nature explorer and photographer. Some of the species that have been shared in the book that we are to publish, the jewels of orchids in Terengganu. Okay. So some of the dendrobiums in Terengganu, vanilla in Terengganu, and wild species of orchids in still wetlands. And conservation initiatives. Okay, this is just brief what we have done for initiatives. Rescued the orchids from the logging concessionaries. Okay, so this is not something that can be done easily, but it's need a lot of effort and also a lot of persistency and rescued orchids, and it has been kept in the wild orchids of Terengganu ex situ conservation in Kuala Terengganu. Okay, so these are things that are being kept. Okay, and apart from that, the initiative, we publish books, okay, by the UPM team together in collaboration with Jabatan Hutan, the forest departments of Sarawak, the forest departments of Perlis, forest department of Selangor, as well as JPSM. Okay, some of the books, and as well as one of the book has been published together with the MBSA, Majlis Bandaraya Shah Alam. So this is a new book that is being prepared or have been published. Okay, so some of the highlights of the papers that we actually publish in, we are called to publish a paper in Orchid Review. And for your information, for Orchid Review, you cannot just send your manuscript. You must be invited, then only you can publish your articles. So this is one of the initiative as well, one of the initiative by Selangor Forest Department. They create a conservatory, wild orchid conservatory in Fraser's Hill. The Fraser's Hill is basically not only consists of Pahang, but it's actually shared with Selangor and also Pahang. So this is created or in the part where that belongs to Selangor. Okay, some of the books and so on. Then apart from during this pandemic, we cannot just say that it's pandemic, so we just relax at home. No, we are not supposed to do that. For me and my team, we persistently writing papers and publish and send papers to journal. Rejection upon rejection, we continue and persist to send to any journals that we think that's suitable to highlight all our findings. So so far we have, this is only part of it, is we have published 13, 13 papers since September last year. Okay, and the books I have published, I have written 4 books so far in 2020 to 2021. So this is the new books that the Shah Alam City of Orchids, and also the Magnificent Wildflowers of Fraser's Hill. So these are the 2 books that you will see soon be made available. It's in the process of printing. We can't print the book because we are under NCO and the publisher cannot open their factory. So therefore, this has been delayed for the past 6 months. So with that, I want to just share with you that in order to make something beautiful, right, you have to have your character, intelligence, strength, and style, and that makes beauty. And in orchids, you can always find this. And I think that those people who works with flowers as well as with orchids and all biodiversity and loving nature, for example, will be forever young, like Prof. Ahmad. You may see that he has white hair, but that white hair is actually glory for his, it's the glory that covers his head. Okay, with that I said thank you.
Okay, thank you, Rusea. The last sentence I couldn't hear it because something dropped near me. Okay, thank you. One thing good about this is you can go to the forest, you can go to Leiden, you can go to Kew, or many other places. Sometimes you have to divert the focus a bit. Instead of study science and biodiversity, what make you travel all over the world free? You know, so I always give example like bats. When the bats fly, then you can fly also to other places, so that you can study about bats. And we can give example. Those who study about bats can travel all other places in the world because bats are an issue. Same thing with the orchid. It's not just looking at the biodiversity in the forest, but we also look on the effect of climate change, and others at the same
Yes.
time is opportunity for the business. So right, I can see many people, but unfortunately we don't have enough time. One question for Dr. Ain is about, in general, I can say, is that a specific, the Fusarium, the specific to plants at a specific location, or you can find easily everywhere, Dr. Ain? Because I have the problem with Fusarium in my house now. Where are you? Yeah, Dr.
Okay.
Ain.
Yeah, I'm here. So, okay, actually, if the Fusarium is non-pathogenic Fusarium, so actually they have no host specificity unless that they are pathogenic. For example, that I give Fusarium oxysporum for Specialis cubense, they only pathogenic to banana, and others, for example, that Molinis pathogenic to melanine. So yes, many fungi associate with many plants and live in a specific environment. So for example that but they are diverse. For example, that Fusarium semitatum, actually you can find them all over area by the sample. For example, if let's say that you have here that you mentioned about the content area, for example, actually you can find the Fusarium semitatum. So actually if they are non-pathogenic, so means that they are diverse and you can find in various habitat as well.
Why they have a problem with say banana or other fruits? Is it because of they don't have the the enemy?
I don't
Yeah, I
get
mean,
your
is there
question.
any other organism that can kill him or tell them when they are exist? You know, if you go to the forest, you see in the forest is quite stable, but it's sometimes when we have unstable environment, the imbalance of the, say for example, fungus or microbe, then you can see the problem with the plants or whatever parasite.
So actually based on the nature lah. Usually if the fungal are dominant, so usually the others, because of Fusarium is rapid growth, same as Aspergillus and Penicillium. Usually when they colonize that area, so the others fungi that is slow growth usually cannot cope and colonize the area. So that's the reason if they are pathogenic, so because of they are rapid growth, so they will colonize that area lah. So for example that so because of the disease, the three factors about the organisms, the pathogens, the host, and the environment. So if all these three that's suitable for this organism can cause a disease, so they will dominance and colonize that area. So that's the reason the other fungi, the other organism, even the bacteria, the virus, you cannot actually control them. So unless some of the factors, for example, they are pathogenic but the host is resistant, so they will not cause a disease lah. So it depends on these three factors lah. So yeah, and depends on the growth lah.
Yeah, that's good challenge, because if they live for last 20 years, it's okay, but I'm very sure they have many hundreds years ago. So if they are truly rapidly growth and no pathogen, no whatever other animal that can stop them, I think the world will be full of fungi. So this statement is challenge the young scientists to fight against it. You know, we sometimes we found it in the test tube, but in the real world it's different behavior. This is what we want to see for the future balance in life. Because I always said why in the natural forest we don't put fertilizer and we don't put the pesticide. So when Prof. Rusea, for example, will bring back the orchid and then they grow it and then they start putting fertilizer and pesticide. In the forest, they are growing well at a certain location, at a certain climate, and they are growing very well. So this is a challenge for the future scientists, for young scientists to look for the balance of the ecosystem. And when you talk about sustainability, but at the same time you're doing something else, like what people are doing now, roadmap to single-use plastic, but every day they donate ton of plastic to people. So it's a conflict, you know. This is a challenge for the young generation to solve it. Thank you, Dr. Ain. Any other question? So I would like to
Sorry,
thank, yeah, siapa tu?
maybe I give last words that actually
Okay,
because
right.
of
Last sentence, yeah.
so yeah, because of the organism then cause disease only 3%. So we still safe. That's we have 97% that as a saprophyte, and also they are very good organism. So yeah, okay. So thank you.
Okay, right. Thank you very much. We need to look for it. We don't want 3% of organism create life, a problem in our life. So this is what we want to make public understand, and they want to see this little percentage, and they're so excited to put more fertilizer on it, and whereas we can find other ways to solve the problem. This is the issue that the biologists and the ecologists should see. One of the purpose of this series of seminar that we're going to have is we want to look at this solution for the future generation. So thank you, Dr. Ain. Thank you, Prof. Rusea. I know you are busy, but you have time with us today. Hopefully, this is not the first one. That's why I planned for 20 minutes, because we have to have a few series later. We call you back because when we find the issue right, and all of us should come out and contribute. And today we got 60 or 70 people are watching and listening, and many more say, sorry, I cannot come. And it shows that more than 100 people actually attracted to our seminar series. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.